Hello everyone, a very welcome to Vyas Edification Kota. So today we are starting with a new chapter in the series of NCERT discussion here, and the chapter is sexual reproduction in flowering plants. So we are going to discuss each and every question given in your NCERT book in this chapter, that is sexual reproduction in flowering plants, and this chapter is from class 12, which is the second chapter of that book. And also this chapter contains total 18 questions in it. Now in this session, in this series, we are going to discuss all of the questions given in your NCERT one by one in detail. So let's begin with the very first question of this chapter, that is, name the parts of an angiosperm flower in which development of male and female gametophyte takes place. So first of all, what is gametophyte? We must know what is gametophyte. Gametophyte is any structure which is going to give rise to gametes. Any structure which is going to give rise to gametophyte is known as gametes is known as gametophyte. Now here we comes with what is male gametophyte and what is female gametophyte in case of flowering plants or angiospermic plants. So we know that in case of male we have pollen grains. Pollen grains are not male gametes. People often confuse with the pollen grain as gametes or gametophyte, but it is not gamete. Pollen grains are actually gametophyte. So in case of male, in case of angiospermic plants, we have male gametophyte as pollen grain, and in case of female, in angiospermic plants, we have embryo sac as female gametophyte. Ovule, remember, ovule is not a gametophyte, it is a megasporangium, inside which the female gametophyte known as embryo sac is being developed. So now we know that what is gametophyte. Any structure which is going to give rise to gametes is known as gametophyte. Gametophyte can be of two types, that is male gametophyte and female gametophyte. In case of angiospermic plants or flowering plants, as we are studying about the sexual reproduction in flowering plants, so in case of angiospermic plants or flowering plants, we have male gamete as pollen grains and we have female gamete as embryo sac. Now we have to discuss where does the development of these two gametophytes, the gametophyte such as pollen grain and the gametophyte such as embryo sac, both these two these two are the gametophytes. Where does the development of these two gametophytes actually takes place? So if we discuss first of all the development of a male gametophyte, then we know that in case of angiospermic plants or flowering plants, we have flowers as their reproductive organs or reproductive structures, inside which the male and female reproductive organs or reproductive parts are present. If we talk about the male reproductive organ, that is stamen, which is consisting of two parts, that is anther and filament. Now anther is bilobed and both of the lobes, each lobe of the anther contains two microsporangias which are the sac-like structures. Alright, so in total we have 4 microsporangias in anther. Inside microsporangias, we have a special tissue known as porogenous tissue and a special kind of cell known as pollen mother cell or microspore mother cell. These pollen mother cells or micro mother cells being diploid in nature as they are part of the flowering plants, they undergo meiotic divisions to form pollen grains. They undergo meiotic division as they are diploid in nature. They undergo meiotic division to form a male gametophyte known as pollen grain. So now we know that where does the pollen grain or male gametophyte develops? Inside the anther, inside the microsporangias in anther, the pollen grain or male gametophyte actually develops. If we talk about the female gametophyte, then the female gametophyte development takes place inside the megasporangium. Now, what is female megasporangium? The ovule is the female megasporangium. Now, as we know that in flowering plants or angiospermic plants, the reproductive structure, total reproductive structure is flower, which can contain both male and female reproductive organs. In case of female reproductive organ, we have pistil or carpel. This pistil or carpel, being female reproductive organ, consists of 3 parts. It is made up of 3 parts such as stigma, style and ovary. Inside ovary, there is a cavity known as ovarian cavity or locule. Inside this locule or ovarian cavity, we have ovule as a megasporangium, which is attached to placenta with the help of a funicle. Inside this ovule, which is also known as female megasporangium, we have a special kind of tissue, that is nucellus tissue, and a special kind of cell known as megaspore mother cell. These megaspore mother cells are also diploid in nature as they are part of the flowering plant. They also undergo meiotic divisions to form female gametophyte known as embryo sac. So now we have completed that where does the development of male gametophyte and female gametophyte takes place. In case of pollen grains, it develops inside the microsporangiums in anther, and in case of female gametophyte, this develops inside the female megasporangium known as ovule. So now if we want to write the answer, then we know the answer of what is gametophyte. Any structure which is going to give rise to gamete is known as gametophyte. But the question is asking where does the male gametophyte and female gametophyte develops in case of angiospermic flower. So first of all, we will write the male gametophyte. The male gametophyte such as pollen grain. The pollen grain is male gametophyte. So the male gametophyte such as pollen grain develops inside the microsporangias and these microsporangias are present in anther, which is the part of a stamen. And what is stamen? Stamen is actually a male reproductive organ or male reproductive part in case of angiospermic flower. Now similarly, the female gametophyte, what is female gametophyte? The female gametophyte is not ovule, it is embryo sac, such as embryo sac. So the female gametophyte such as embryo sac develops inside the female megasporangium, inside the female megasporangium. Now what is female megasporangium? What is female megasporangium? The female megasporangium is an ovule. So the development of pollen grains, which are known as male gametes, male gametophyte, they develop inside the anthers. Where in the anthers? Inside the microsporangia of anthers. And in case of female gametophyte, the development of female gametophyte such as embryo sac takes place inside the female megasporangium, which is known as ovule. So this is the complete solution of question number 1 here. Now we have completed it. Now here we comes with question number 2, and the question number 2 is asking differentiate between microsporogenesis and megasporogenesis. Which type of cell division occurs during these events? Also, the third part of this question contains name the structure formed at the end of these two events. So basically this question number 2 contains 3 parts in it. The first part is differentiate between the microsporogenesis and megasporogenesis. The second part is what type of cell division actually occurs during all these two events. And the third part of this question contains what are the structures formed at the end of these two events, such as microsporogenesis and megasporogenesis. So let's begin with the answer of question number 2. First of all, we will answer the first part of this question, that is, what is the difference between microsporogenesis and megasporogenesis? We have to differentiate between them. So first we are going to discuss the microsporogenesis. Microsporogenesis is actually a process of formation of haploid gametophyte, haploid gamete from diploid pollen mother cell, or we can say diploid microspore mother cell. So microsporogenesis is actually a process of formation of haploid gametophyte from diploid pollen mother cell or microspore mother cell. Now, if we talk about the megasporogenesis, megasporogenesis is also a process of formation of haploid female gametophyte from diploid megaspore mother cell. So these are the two events, these are the two processes occurring in the different parts of a flower which will give rise to male gametophyte and female gametophyte. In case of microsporogenesis, as the name suggests, the process in which the microspores are going to be formed. Microsporogenesis is a process in which the microspores are going to be formed. What are these microspores? These microspores are nothing but pollen grains. These pollen grains are going to be formed from diploid pollen mother cell or microspore mother cell. So this whole process is known as microsporogenesis. In case of megasporogenesis, the formation of megaspore is going to be takes place. What is the megaspore? Megaspore. That megaspore is embryo sac. This embryo sac is going to develop from diploid megaspore mother cell. So these all two the processes, and this is the difference between microsporogenesis and megasporogenesis. Microsporogenesis is a process which occurs in the male reproductive organ of a flower and will give rise to microspores such as pollen grain. Megasporogenesis is a process which occurs inside the female reproductive organ, which is going to give rise to female megaspore such as embryo sac. So this is the difference between these two events, these two processes, that is megasporogenesis and microsporogenesis. Now we have to tell what type of cell division occurs during these events. So if we discuss in the microsporogenesis, which occurs in the male reproductive organ and is going to give rise to a male gamete or gametophyte or a male microspore known as pollen grain, we know that where does the development of these pollen grains, microspores or megaspores takes place? Inside the reproductive organs of their respective structures. Alright, if we talk about the microsporogenesis, then we know that stamen is actually a male reproductive organ which consisting of two parts, that is anther and filament. Inside the anther we have microsporangia. Inside microsporangia we have a special tissue known as sporogenous tissue and a special kind of cell known as microspore mother cell or pollen mother cell. These pollen mother cells present inside the microsporangia or pollen sac are diploid in nature. These pollen mother cells or microspore mother cells undergo meiotic division. So this is the division, this is the type of division which occurs in the event of microsporogenesis. What kind of division is taking place? Meiotic division is taking place to form a male microspore, that is pollen grain. So the pollen mother cells present inside the pollen sac undergoing meiotic division to form haploid microspores known as pollen grains. Whereas if we talk about in megasporogenesis, we know that megasporogenesis takes place inside an ovule which is present in the locule or ovarian cavity of a female reproductive organ. Now, we know that a special kind of tissue, that is nucellus, and a special kind of cell known as megaspore mother cells are present inside the ovule. These megaspore mother cells are also diploid in nature, and being diploid in nature, they also undergo meiotic division to form haploid embryo sac or female megaspore. So both in the events of microsporogenesis and megasporogenesis, the division, the type of division taking place is meiotic division, which is also known as reductional division. So the reductional division or meiotic divisions are the type of divisions which are taking place during the process of microsporogenesis and megasporogenesis. Now here we comes with the third part of this question: what structures are formed at the end of these two events? So at the end of these two events, such as microsporogenesis, at the end of the process of microsporogenesis, we will get pollen grains, which are the male gametophyte, alright, or we can say microspores. And at the end of a process of megasporogenesis, we will get female gametophyte such as embryo sac, or we can call it megaspore. So now we have completed, and let's write the answer that first we will differentiate between the megasporogenesis and microsporogenesis. So first I am writing about the microsporogenesis. Microsporogenesis, a process of formation of haploid microspore from diploid pollen mother cell, or we can call it microspore mother cell. Where these pollen mother cells or microspore mother cells are present, actually they are present inside the microsporangias or pollen sac. So a process of formation of haploid microspore from diploid pollen mother cell present inside the microsporangia or pollen sac, inside the microsporangia or pollen sac. This process is known as microsporogenesis. Now in case of megasporogenesis, if we want to write megasporogenesis, then this is also a process of formation of megaspore, a process of formation of megaspore or a haploid megaspore. Haploid because the megaspores are actually haploid, because they are formed from the meiotic division. So always these gametophytes or microspore or megaspore, all these are the spores which are haploid in nature. So a process of formation of haploid megaspore from a diploid, from diploid megaspore mother cell, megaspore mother cell, which is present inside the ovule, which is present inside the ovule, which is female megasporangium. So this is the difference between microsporogenesis and megasporogenesis. Now if we want to write what kind of divisions take place or taking place during these two events, so if we want to write the second part of this answer, question, then pollen mother cells undergo meiotic division to form haploid microspore. So what kind of division you are seeing in microsporogenesis? That is meiotic division in case of microsporogenesis, and in case of megasporogenesis, megaspore mother cells also undergo meiotic division, which is reductional division. Alright, these two events are taking place through reductional division. So megaspore mother cells also undergo meiotic division to form female gametophyte or megaspore. Alright, so inside or during the event of megasporogenesis, what kind of division is being seen? Also in this we will see the meiotic division. Meiotic division takes place in megasporogenesis or microsporogenesis. Both in the events of microsporogenesis or megasporogenesis, the division type is actually reductional division or meiotic division. Now comes towards the last part of this question. Last part of this question contains what are the structures which formed at the end of these two events. So at the end of microsporogenesis, the pollen grains are going to be formed. All right. Now, and at the end of megasporogenesis, megasporogenesis, the embryo sac is going to be formed. So now we know that at the end of microsporogenesis, pollen grains are going to be formed, and at the end of megasporogenesis, the embryo sac is going to be formed. Now we have completed the solution of question number 2 over here. Now let's come towards the question number 3. Arrange the following terms in correct developmental sequence, and some terms are given over here: pollen grains, sporogenous tissue, microspore tetrad, pollen mother cell, and male gametes. So we know that these all terms given on the screen are actually related to male gametes or the formation of male gametophyte, which is known as microsporogenesis. Now what is microsporogenesis? We already know that microsporogenesis is actually a process of formation of haploid gametophyte or haploid microspores such as pollen grains with the help of meiotic division or reductional division. these pollen grains or microspores are going to form the male gametophyte, which is known as pollen grains. Now, if we talk about the microsporogenesis, the sequence, the developmental sequence of this would be, first of all, the sporogenous tissue. Sporogenous tissue is the first because it is present inside the microsporangias or pollen sacs. Then after sporogenous tissue, we will have pollen mother cells which are going to give rise to microspores. So these pollen mother cells are diploid in nature. The sporogenous tissue is also diploid in nature. These pollen mother cells being diploid in nature, they will undergo reductional division or meiotic division to form microspores or pollen grains. So after pollen mother cells, what will they do? Undergoing meiotic division, it will form microspore tetrad. It will form microspore tetrad. That means 4 set of cells. One pollen grain or one microspore undergoes meiotic division, will be forming a tetrad. A tetrad, which is a 4 set of microspore cells, and each of the microspore is actually develops into pollen grain. So after microspore tetrad, we will get pollen grain. Inside pollen grains, there is 2 cells are present. There are 2 cells are present, that is generative cell and vegetative cell. These generative cells inside pollen grains are going to give rise to male gametes. So after pollen grains, we will get male gametes. So this is the sequence, and if you want to write the answer, then we know that microsporogenesis. Answer will be like this: microsporogenesis is the process of formation of a haploid microspore, such as pollen grain. What are the male microspores which are haploid in nature? That is pollen grain. So microsporogenesis is the process of formation of haploid microspores such as pollen grains with the help of meiotic division or reductional division. Now after undergoing meiotic division, this microspore tetrad is going to give rise to pollen grain, and finally the pollen grain is going to give rise to male gametes. So the sequence, the developmental sequence is as follows. Now we will write the sequence of all these terms given over here. So first will be sporogenous tissue. The number 1 will be sporogenous tissue. After the sporogenous tissue, we will get pollen mother cell, which is diploid in nature and present inside the microsporangias or pollen sac. Now after pollen mother cell, on the 3rd number, we will get, as these pollen mother cells are diploid in nature, they will undergo meiotic division to form microspore tetrad. So we will get microspore tetrad. After microspore tetrad, what we will get? After microspore tetrad, each of the microspore of tetrad will develop into pollen grain. So will develop into pollen grain. On 4th number, we will have pollen grain, and at last, after the development of pollen grain, we will get the male gametes, as these male gametes are going to be formed from pollen grains. So this is the complete solution of question number 3. Now let's come towards the question number 4 with a neat labeled diagram. Describe the parts of typical angiosperm ovule. So in this question we have to draw a neat labeled diagram of an ovule, and also we have to describe all the parts in detail of it. So first of all, what is ovule? Ovule is actually an integumented megasporangium. Ovule is actually an integumented megasporangium, and in case of angiospermic plants, the ovule is enclosed within an ovary. The ovule is present inside the ovarian cavity in case of angiospermic plants, whereas the ovule is not enclosed by an ovary in case of gymnospermic plants. That is why the seeds are called as naked seeds, or the plants is termed as naked seeded plants, because the ovule is not enclosed within any ovary in case of gymnospermic plants. But in angiosperms it is enclosed within an ovary. An ovule is actually a female gamete, female megasporangium. If we talk about the ovule, inside the ovule we have a special kind of tissue known as nucellus, which have food reserve material, abundant food reserve material. So inside an ovule we have a special kind of tissue known as nucellus, which is abundant food reserve material, which has abundant food reserve material. Also ovule have some coverings which are known as integuments, which protect the developing embryo, and also a special kind of cell that is known as megaspore mother cell, which is diploid and going to give rise to megaspore. Now if we draw a diagram of ovule, it will be like this. Now, if we label, first of all the diagram is of ovule, and if we label, then this is the point known as hilum. This is the point known as hilum. This is the point known as funicle. This is the point which is known as funicle. This is outer integument. Outer integument. The inner one is inner integument. These are the coverings of an ovule. This region where the integuments are actually not covering the ovule is actually a region known as micropyle region. This is micropyle. Now inside which, after you can see inside the integuments, this is the tissue, this is the nucellus tissue. Nucellus tissue which has abundant food reserve material for the developing embryo, and this one, the innermost structure inside the ovule, you can see is embryo sac. This is embryo sac, and opposite to the micropyle region, the region here is known as chalazal and or chalazal region. Chalazal and or chalazal region. So this is the complete labeled diagram of an ovule, which is female megasporangium. Now we have to describe all these parts in detail. We know what are the parts of ovule. Hilum is one part. Then we have next part as funicle, micropyle region, in outer and inner integuments, the nucellus tissue and embryo sac, which is a female gametophyte. Female gametophyte, which is going to give rise to female gamete inside it. Alright, now we will be going to discuss each and every part in detail. So first, First, if we talk about the very first part, that is hilum. What is hilum? Hilum is actually an attachment point. It is an attachment point where the funicle attaches to the body of an ovule, to the body of an ovule, Now, the second structure is funicle. What is funicle? Funicle is the attachment structure which attaches the ovule with placenta within the ovary. So, what is funicle? It is the attachment structure, stalk-like structure actually, stalk-like structure which joins the ovule, with placenta, inside the ovarian cavity or locule. Then the third part we have is integuments. What are the integuments? These are the covers, these are the coverings which protect the developing embryo. So, integuments are the coverings which protects the developing embryo. The next part in ovule structure we have is nucellus tissue. So, what is nucellus tissue? Nucellus tissue is a special kind of tissue. It is a special kind of tissue present inside the ovule, which has abundant food reserve material. So, what is nucellus tissue? Nucellus tissue is a special kind of tissue which is present inside the ovule, and it has abundant food reserve material for the nourishment and development of developing embryo inside the ovule. Last but not least, the fifth one is embryo sac. Which is also known as female gametophyte. This is also known as female gametophyte. This female gametophyte is going to give rise to female gamete inside it. So, now with this we have completed the solution of question number 4 over here with the neat and labeled diagram of an ovule, and also we have described the parts of an ovule here. So, now let's move on to the next question, that is question number 5. What is meant by monosporic development of female gametophyte? Now, here the question is asking, what do you mean by monosporic development of female gametophyte? So, first of all, megaspore is the first cell. Megaspore is the first cell of female gametophyte, and depending on the number of megaspore, the female gametophyte is termed as monosporic, bisporic, or tetrasporic. Alright, this megaspore, the megaspore, which is the first cell of female gametophyte, undergoes meiotic division or reductional division. This megaspore undergoes meiotic or reductional division to form a dyad and then tetrad of megaspore. This megaspore undergoes meiotic division to form dyad and a tetrad of megaspore. Among these four set of cell, which is known as tetrad of megaspore, one megaspore becomes functional or it remains functional and three among the tetrad gets degenerated. That one functional megaspore undergoes three simultaneous mitotic division or equational division to form 2, 4 and then 8 nucleate and 7 cell structure, which is known as embryo sac. This embryo sac is actually known as the female gametophyte. So this is the answer, and if you want to write the answer of question number 5, it will go like this. That first of all, megaspore is the first cell, of female gametophyte. Why this is known as the first cell of female gametophyte? Because from this particular cell, because from this megaspore mother cell, only this cell undergoes meiotic division to form female gametophyte further. Now, depending upon the number, of megaspores, the female gametophyte is termed as, it can be termed as monosporic if the whole female gametophyte is developing from one megaspore, then it could be named as monosporic development of female gametophyte. It could be bisporic if it is developing from 2 megaspores, and it can be tetrasporic. It also can be tetrasporic in case of angiospermic plants. So it can be monosporic, it can be bisporic, and it can be tetrasporic depending on the number of megaspore present in the ovule. So now what happens? This megaspore mother cell undergoes meiotic division. It undergoes meiotic or reductional division. To form, what it will form? To form first dyad, then a tetrad of megaspore. First a dyad and then tetrad of megaspore. What kind of division will take place to form a dyad and tetrad of megaspore? As we know that in the process of megasporogenesis, megaspore mother cell undergoes meiotic division or reductional division to form a megaspore tetrad. That means 4 set of cell. Alright, a set of cell which have 4 cells, and all these 4 cells are known as megaspores. So now this undergoes meiotic division or reductional division to form diad and then tetrad of megaspore. Among this tetrad, one megaspore remains functional, and all other than this three megaspores will degenerate, and this functional megaspore undergoes three mitotic divisions, three mitotic or equational division, to form what it will form? To form 2, 4, then 8 nucleate 7 cell structure. 2, 4, then 8 nucleate and 7 celled embryo sac, which is also known as the female gametophyte. So now I hope you are clear with the answer of question number 5, that what is monosporic development. If any if any female gametophyte is developing from only single cell, then the development of female gametophyte is known as monosporic development. And now this is the complete answer, complete solution of question number 5 over here. So now let's come towards the question number 6. With a NEET diagram, explain the 7-celled 8-nucleate nature of female gametophyte. Now we know that what is female gametophyte? Female gametophyte is embryo sac. Embryo sac is the female gametophyte in case of angiospermic plants. Now this embryo sac is actually 8 nucleated 7-celled structure, which is developed from only one megaspore mother cell present in the nucellus of an ovule, which is a female megasporangium. So this female gametophyte known as embryo sac is developing inside the nucellus tissue of an ovule, which is female megasporangium. Now if you talk about the female gametophyte, that is embryo sac, what does it contain? Or if we want to describe the structure of embryo sac or female gametophyte, then it would be as follows. Embryo sac, the female gametophyte consisting of 8 nuclei and 7 cells. Basically, I must say that the embryo sac is actually formed of 8 nuclei and 7 cells. This 8 nuclei and 7 cells are constituting together to form an embryo sac, which is known as female gametophyte. Now, what are these cells or what are these nuclei called inside the embryo sac? We know that ovule is an microsporangium. Ovule is an integumented megasporangium which have nucellus tissue, megaspore mother cell and integuments. We know that there is a region where integuments are not covering the ovule. That particular region in the ovule is known as micropillar end. And opposite to the micropillar end or region, there is another region known as chalazal region. So if we talk about embryo sac, the cells which are present at the micropillar end in the embryo sac are known as egg cell, and synergid cells. So basically at the micropillar end, the three cells are constituting together to form an egg apparatus. Egg apparatus is consisting of one cell which is forming egg cell, and along with the egg cell there are two other cells are present, that is synergid cells. These two synergid cells have finger-like projections on their walls, and this is known as filiform apparatus. Alright, this all three set of cells, egg cell and two synergid cells forming egg apparatus. Now on the opposite side of micropillar end there is chalazal end, and on the chalazal end we have all three set of cells, and all these three cells are known as antipodal cells which will degenerate after fertilization. Now in the middle of the embryo sac, which is known as central cell, there are two nuclei present. Two nuclei are present and which are known as polar nuclei. They fuse together and form secondary nucleus, and after the fertilization they will transform into endosperm. So now this is the explanation, and I am going to draw a diagram of a female gametophyte over here. So let's just make female gametophyte. This is an embryo sac. Alright. These are the integuments, and is a region here where the integuments are not covering the ovule, where the integuments are not covering the ovule. That particular region is known as micropillar end. So here now we have a diagram of this embryo sac. Now let's just label it once. First of all, this region where the integuments are not covering the ovule, this region is known as micropillar. Micropyle region I can write. Micropyle region. Alright. As I said that at the region of micropyle there is an egg apparatus consisting of three cells. So I will make three cells over here. These are the three cells. One cell is here. This one cell along with the nuclei, of course each cell will have nucleus. So this one cell in the middle is known as egg cell. It is egg cell which is the female gamete, and along with this egg cell there are two cells present. These two cells are known as synergids. These two cells are known as synergids. So these are synergids. Alright, and these two synergids plus one egg cell are forming egg apparatus. So this whole thing is known as egg apparatus. Egg apparatus. Alright, now all these two synergid cells have finger-like projections over here on the wall of them. These finger-like projections on the wall of synergid cells is known as filiform apparatus, which guides the pollen tube, which guide the formation of pollen tube inside the embryo sac. So this is filiform apparatus. Now, as I said that opposite to the micropyle region there is another region known as chalazal region. So this is chalazal region. This is chalazal region, and on the chalazal region we have three cells. We have three cells, of course with nuclei. All these three cells are known as antipodals. All these three cells are known as antipodals, which will get degenerate after fertilization or during fertilization. Now this whole thing between this region and this region is known as central cell. This whole part is known as central cell. Central cell, which consisting of two nuclei, These are not cells, these are two nuclei. These two nuclei are known as polar nuclei. Alright, these two are known as polar nuclei. So we have completed the structure with labeling of female gametophyte. Female gametophyte is embryo sac. Now let's talk about the description or explanation of all these parts. Cells or egg apparatus present at the micropillar region of an embryo sac. Alright, so this egg apparatus, as I already mentioned, that egg apparatus is consisting of 3 cells, 2 synergid cells and 1 egg cell. This one egg cell is actually a female gametophyte. This is female gametophyte. Alright, this is female gametophyte. So, female gamete. This is female gamete. This whole embryo sac is female gametophyte. So this is egg cell, and egg cell is known as female gamete, which is going to fertilize with male gamete to form a zygote over here. These two cells present near the egg cell are known as synergids. Alright, synergids. These two synergid cells have finger-like projections on the wall of them. These finger-like projections are known as filiform apparatus. Now what is the function of this filiform apparatus? They guide the formation of pollen tube through pollen grain. Alright, so they guide the formation of pollen tube. Egg cell will fertilize with male gamete to form zygote. Then we have antipodal cells on the chalazal region. These antipodal cells will get degenerate. These antipodal cells will get degenerate after or during the fertilization. And now we are remaining with the 2 nuclei present in the central cell of an embryo sac. What happens before fertilization? Just before fertilization, these 2 polar nuclei fuses with each other. These fuses with each other to form secondary nucleus. This secondary nucleus will fertilize with another male gamete, as we know that a single pollen grain is consisting of 2 male gametes. A single pollen grain is capable of making or synthesizing 2 male gametes at a time. So among all these 2 male gametes, 1 male gamete will fuse with egg cell, and the remaining 1 male gamete will fuse with the secondary nucleus present in the central cell. After the fertilization or triple fusion here, as the 2 nuclei of female gametophyte and 1 nucleus of male gametophyte are going to be fused with each other, we will call it as triple fusion, as 3 nuclei are fusing with each other. So triple fusion, the resulting of triple fusion, we will get endosperm. We will get endosperm, which is going to provide nutrition or nourishment to the developing embryo from this zygote over here. So this is the explanation and a diagram with proper labeling of a female gametophyte, and this is the complete solution of question number 6. Now let's talk about question number 7. What are chesmogamous flowers? Can cross-pollination occurs in cleistogamous flowers? Give reason for your answer. So first of all, we should know that what are the chesmogamous flowers and what are cleistogamous flowers. So first of all, if we talk about the what is chesmogamous flowers, chesmogamous flowers are the flowers which have exposed anthers as well as exposed stigma into the environment, because they opens up at their maturity. So these kind of flowers, when they become mature, they will have exposed anthers and exposed stigmas into the environment. These flowers are known as chesmogamous flowers. Whereas if we talk about the cleistogamous flowers do not have exposed anthers and stigma into the environment, because they do not open at all at their maturity. When they become mature, some flowers are there, when they become mature, they do not open at all. So cross pollination cannot occur in them because they do not open at all at their maturity. Hence, the anthers and stigma are not exposed into the environment. So, there is no chance of cross pollination can occur or take place into the cleistogamous flowers. Cleistogamous flowers will only have self-pollination, whereas chesmogamous flowers always follow cross-pollination. However, in cleistogamous flowers, seed production takes place, and this seed production takes place with the help of self-pollination only. There are some plants which have both type of flowers in it, such as oxalis, viola. These are the example of some plants which constituting all two types of flowers on its single body of plant, that is cleistogamous flowers and chesmogamous flowers. But why is it so? These plants consisting of two types of flowers on their body provides them cross pollination chances if they skip the chance of cross fertilization or cross pollination, then they will produce seeds with self pollination for sure. So this is the answer, that what is chesmogamous flowers and what is cleistogamous flowers, and why do cross pollination can happen in cleistogamous flowers, and if not, then why the cleistogamous flowers cannot have cross pollination. So if you want to write the answer, then first of all we will write what are chesmogamous flowers. So the chesmogamous flowers are the flowers with exposed anthers and stigma into environment as they open up at their maturity. So chesmogamous flowers are the flowers which have exposed anthers and stigma into the environment at their maturity, whereas cleistogamous flowers cleistogamous flowers will not have exposed anthers and stigma, as they do not open up at their maturity. When they do not open up their maturity, their anthers and stigma will not expose into the environment, so they do not open up at their maturity. Alright, and because they do not open up at their maturity, hence they do not have exposed anthers and stigma into the environment, there is no chance. So therefore they do not have a chance of cross fertilization, cross fertilization or pollination or cross pollination. However, they produce a seed. They produce seed They produce seed with the help of self-pollination. Self-pollination. They will undergo self-pollination to produce seeds because they are not having exposed anthers and stigma into the environment. So there is no chance of cross-fertilization or pollination in them. So they will produce seed with the help of self-pollination. And this is the complete solution of question number 7. So let's talk about question number 8. Mention 2 strategies evolved to prevent self-pollination in flowers. So we have to mention some strategies over here which have evolved in plants to prevent self-pollination in flower. So we have given many strategies to prevent self-pollination in our NCERT book, and among all these strategies we will discuss over here 2 strategies among them. The first is self-incompatibility and the second one strategy is known as proto-entry or proto-gyne. So the first strategy, the self-incompatibility, is actually a condition basically in which the pollen is ineffective of fertilizing the same flower or other flower on the same plant due to inhibition of growth of pollen tube in stigma and style of the female reproductive organ, which prevents the delivery of male gametes to the ovule. So this is one of the strategy which is evolved in plants to prevent self-pollination in flower. The another strategy which is evolved in plants to prevent self-pollination is proto-entry or proto-gyne. Now these strategies are known as the maturation, where the maturation of one reproductive organ becomes mature before another one becomes mature. In proto-entry, what happens? In proto-entry, the maturation of stamen, the maturation of male reproductive organ becomes first, and then after that the female reproductive organ gets mature. In case of proto-gyne, the maturation of female reproductive organs such as pistil or carpel becomes before the maturation of stamen. So this is the 2 strategies. These are the 2 strategies to prevent self-pollination, and let's write the answer. The 2 strategies to prevent self-pollination are as follows. So first one we are going to talk about is self-incompatibility. Incompatibility. A condition. It is a condition wherein the pollen is ineffective of fertilizing the same flower or another flower of same plant due to the inhibition in the growth of pollen tube in stigma and style of the female reproductive organ. Pollen tube in stigma and style which prevents the delivery of male gametes, which prevents the delivery of male gametes into the embryo sac or ovule. Basically it will go to embryo sac because inside the embryo sac the egg cell is present. So self-incompatibility will prevent the delivery of male gametes into the embryo sac. The second one strategy which prevents the self-pollination in flower is protoendry. This is a condition wherein the stamen becomes mature before the maturation of pistil. In this strategy we have another type, that is protogyny. So in this, the maturation of carpel or pistil occurs before the maturation of stamen. Alright, so these are the two strategies which can be helpful in preventing self-pollination in flowers. So this is the complete solution of question number 8. Now let's talk about question number 9. What is self-incompatibility? Why does self-pollination not lead to seed formation in self-incompatible species? So first of all, what is self-incompatibility? We already know what is self-incompatibility. First of all, it is a strategy which has evolved in plants to prevent self-pollination in bisexual flowers. Alright, so it is a strategy has been evolved in plants to prevent self-pollination in them. Now, what do you understand by self-incompatibility, or what do you mean by self-incompatibility? So, self-incompatibility is actually a condition wherein the male gametes are ineffective or are unable to fertilize female gamete of same flower or another flower of the same plant. Alright, so it is a condition wherein the male gamete is unable to fertilize the female gamete of the same flower or another flower of the same plant. This self-incompatibility is actually preventing self-pollination in them. It has been evolved for this purpose only. So it is preventing self-pollination in plants but promoting cross-pollination at the same time. This self-incompatibility in plants or flowers are promoting cross-pollination and they are preventing self-pollination in plants. This self-incompatibility can be found in some plants like orchids, tulip, cabbage, broccoli, etc. So these are the examples of some plants where you can find self-incompatibility. All these plants are having bisexual flowers, even then they are not having self-pollination. Why is it so? Because they have been evolved. The plants like orchids, tulip, etc., they evolved a strategy to preventing self-pollination in them, such as self-incompatibility. Alright, so this is about self-incompatibility. Now let's talk about the second part of this question, that is, why does self-pollination does not lead to seed formation in self-incompatible species? We have various reasons behind this. Some of them are, since we know that male gamete is unable to fertilize the female gamete of the same flower, due to this incompatibility, seed formation in the same flower is inhibited or does not occur. This may be occur due to some genetic factors. This may occur due to some genetic factors, or many times it can occur because the maturation of male and female whorls of a flower takes place at different times. So this is the reason behind why does not the self-pollination lead to seed formation in the self-incompatible species. Now let's write the answer of question number 9 here. First we are going to write what is self-pollination, self-incompatibility. So self-incompatibility is a condition wherein the male gamete is ineffective to fertilize the female gamete. It is a condition wherein the male gamete is unable or inefficient to fertilize the female gamete of the same flower, and also another flower of same plant. Alright, so this is a condition wherein the male gamete is unable to fertilize the female gamete of the same flower or another flower of the same plant. This condition is known as self-incompatibility. So now this self-incompatibility is preventing self-pollination. That is why the plants have evolved this type of strategy to actually prevent self-pollination. So this is preventing self-pollination, but at the same time this is promoting cross-pollination. This is promoting cross-pollination even in the presence of bisexual flower. Even in the presence of bisexual flowers. And this self-incompatibility can be seen in some plants, like if we talk about some examples of this, then we have orchids, tulip, cabbage, etc. Alright, now we will be writing the reasons behind the self-pollination does not leading to the seed formation in self-incompatible species. So here we are writing about some reasons. Now we know that first reason could be, since we know the male gamete is unable to fertilize the female gamete, and due to this incompatibility, the formation in the same flower, the seed formation in the same flower is inhibited. The seed formation in the same flower will not occur because of the self-incompatibility, because the male gamete is unable to fertilize the female gamete. This may occur, also this may occur due to genetic factors. Also this may occur due to genetic factors, and the last reason is many times what happens, many times the maturation of male and female walls, that is stamen and carpel of a flower, takes place at different times, So this is the complete solution of question number 9. Now let's talk about question number 10. What is bagging technique? How is it useful in plant breeding program? So if we are talking about the bagging technique, then bagging technique is a part of a process of artificial hybridization first of all. Now if we are talking about the bagging technique is a technique or a process which involves the covering of stigma with the help of bag. So bagging technique involves the covering of stigma with the help of bag. This process is known as bagging technique. This process of bagging technique ensures pollination with pollens from preferred male parent. Alright. Now if we are talking about the usefulness of bagging technique in plant breeding program, so first of all, this bagging technique is used, this approach is used in artificial hybridization programs. Also, this bagging technique helps in the plant breeding programs as it prevents pollen contamination on the stigma. As a result of this bagging technique, one can obtain superior species as we are making we are fertilizing the pollen, we are fertilizing the female gamete with our desired pollen grain or male gamete. So let's write this question. Let's write the answer of this question. Answer number 10. First of all, what is bagging technique? So bagging technique is the covering of stigma with the help of bag. With the help of bag. This process of covering stigma with the help of bag is known as bagging technique. Alright. Now this process ensures the pollination, with pollens of desired male parent, which is desired. Alright. Now if we talk about the usefulness of this bagging technique in plant breeding program, so let's write about these usefulness. Alright, so first of all, this bagging technique is used in artificial hybridization. It is used in artificial hybridization. Alright, second thing is, it is useful, it is very useful in plant breeding programs. Plant breeding programs. How is it useful in plant breeding programs? Because it prevents contamination. It prevents pollen contamination. Pollen contamination on stigma. Alright, this prevents pollen contamination on stigma, and as a result of this process, one can obtain the superior species. One can obtain the superior species. Alright, so this is the complete solution of question number 9. We have completed what is bagging technique. We have completed that what is actually the bagging technique along with the definition of it, and also we have discussed the usefulness or benefits of this bagging technique. Alright, now this is the complete solution of question number 10. Now we have a question number 11 on the screen. What is triple fusion and where and how does it take place? Also name the nuclei involved in triple fusion. So this question contains 3 parts in it. First we are going to talk about what is triple fusion. Then we are going to talk about where does this triple fusion is actually taking place. Then after, and the last part of this question, we are going to name the structure or the nuclei which is involved in the process of triple fusion. So starting with the first part of this question, that what is triple fusion. Now, going towards the triple fusion, we know that you already have heard about what is double fertilization. Alright, you already have heard about double fertilization. It is a unique feature. Double fertilization is a unique feature of all angiospermic plants. Why is it a unique feature of angiospermic plants? Because this double fertilization does not occur in any kind of plants such as gymnosperms, pteridophytes, etc. And also double fertilization is not a feature of animals as well. So double fertilization only can be found in angiospermic plants. Now this double fertilization involves 2 fusions in it. That is why the name given to it is double fertilization. There is 2 fertilization occurring in that process. What kind of 2 fertilization? One is syngamy and the second one is triple fusion. Syngamy is actually a fusion of male gamete from pollen and a female gamete present in the embryo sac of a female gametophyte. Alright, so this fusion of a male gamete and a female gamete which is going to form a zygote, a diploid zygote, this is termed as syngamy. If we talk about the triple fusion, then triple fusion is a process of fusion of 3 haploid nucleus. 3 haploid nucleus fuses with each other to form a triploid structure. Alright, this triple fusion occurs with the help of the fusion of nucleuses from male gamete and 2 polar nucleus present in the central cell of a female gametophyte such as embryo sac. So the polar nuclei, the 2 polar nuclei present in the central cell of embryo sac fuses with each other to form secondary nucleus. This secondary nucleus fuses with the nucleus of a male gamete and forming a triploid structure known as primary endosperm nucleus. This primary endosperm nucleus, because it is formed from the fusion of 3 haploid nucleus, then it is triploid in nature. So now this is all about the question 11, and now we are going to write the answer of this. First of all, we are going to talk about the triple fusion. Then let's start with double fertilization. double fertilization, the answer will start with double fertilization, because moving on to the triple fusion, we must know that what is double fertilization, because triple fusion is actually a part of double fertilization. So double fertilization is a unique feature, to angiosperms. It is not found in any kind of plants or in any kind of animals as well. So this is a unique feature to all angiospermic plants. This double fertilization includes syngamy and triple fusion All right. If we talk about the syngamy, we are not going to write what is syngamy, as the answer will be about triple fusion only. So syngamy is actually a process of fusion of a male gamete and a female gamete. That process is known as syngamy. Come towards the triple fusion. If we talk about the triple fusion, then triple fusion is a process of fusion of three haploid nucleus, two polar nuclei and a nucleus of male gamete. They fuses together, all these three haploid nucleus of female gametophyte and a male gamete fuses together to form a triploid structure. Alright, triploid structure. If we talk about the second part, where and how does it takes place? So where does it take place? Triple fusion takes place inside the embryo sac, which we call female gametophyte. This triple fusion as well as syngamy, both of these processes, or I may call it double fertilization, as it includes both the type of events such as syngamy and triple fusion. So double fertilization or syngamy and triple fusion are occurring inside the female gametophyte, that is embryo sac. So second part of this question, answer of second part of this question is double fertilization or triple fusion and syngamy occurs inside embryo sac, which is the female gametophyte. Alright, how does it take place? We already know that. How does it take place? There is a presence of egg cell in the egg apparatus inside embryo sac. It fuses with the male gamete. Alright, it fuses with the male gamete. There is two polar nuclei also present in the central cell of embryo sac. We know that this is embryo sac. Alright, this is embryo sac. Three cells are at the chalazal end. These cells are known as antipodal cells. Then if we talk about the micropillar end or micropillar region, we have egg apparatus consisting of egg cell and two synergid cells. Now this big cell, which is known as central cell in the middle, have two polar nuclei. These two polar nuclei are haploid in nature, present in the central cell of an embryo sac. These two polar nuclei fuses with the male gamete, one another male gamete, to form a triploid structure. All the fusion, that is syngamy and your triple fusion, both of these fusions are occurring inside the embryo sac only, which is known as female gametophyte. Now let's come towards the third part of this question, that name the nuclei involved in the triple fusion. Name the nuclei involved in the triple fusion. So the nuclei name is polar nuclei. Nuclei is polar nuclei, and when it fuses with the nucleus of male gametes nucleus, it gives rise to primary endosperm nucleus. It gives rise to primary endosperm nucleus, which is triploid in nature, as it is composed of 3 nucleus, or I may say the fusion of 3 nucleus occurs to form this triploid structure known as primary endosperm nucleus. So the name of the nuclei involved in the triple fusion is primary endosperm nucleus, which is triploid in nature. Alright, so this is the complete answer, this is the complete solution of question number 11. Now let's talk about question number 12. Why do you think the zygote is dormant? The zygote is dormant for some time in a fertilized ovule. So yes, zygote is dormant for some time in angiospermic flowers, but why is it dormant? Why it is dormant? The question is asking why the zygote is dormant for some time in a fertilized ovule. So we know that double fertilization is a unique feature of all angiospermic plants. Double fertilization involves syngamy as well as triple fusion. Syngamy is a fusion of male and female gamete, and triple fusion is a fusion of 3 haploid nucleus, 2 polar nuclei and 1 nucleus of a male gamete. Alright, so double fertilization, which is a unique feature of angiosperms, will lead to the formation, double fertilization. Inside the double fertilization, the syngamy and triple fusion we are talking about. Alright, so this double fertilization leads to the formation of a zygote and primary endosperm nucleus simultaneously. Alright, so this double fertilization leads to the formation of zygote and primary endosperm nucleus simultaneously. This primary endosperm nucleus divides to form a large nutritive endosperm. This primary endosperm nucleus, which is made from the fusion of 3 haploid nucleus, this undergoes division, this undergoes several mitotic divisions to form a large nutritive endosperm for developing an embryo. Alright, this ensures the source of nutrition for developing an embryo. The formation of endosperm, which is a large nutritive tissue, is actually ensures the source of nutrition for developing an embryo. And this ensures, because of this only, the zygote is actually dormant for some time in a fertilized ovule, so that when the embryo gets developed, when the embryo is developing, it will have sufficient amount of nutrition from endosperm. That is why the zygote remains dormant in a fertilized ovule. So now let's talk about, or let's see how we can write the answer of this. What do you think the zygote is dormant? So double fertilization, we will start our answer double fertilization. Double fertilization leads, we know that double fertilization is having 2 processes such as syngamy and triple fusion. So double fertilization leads to the formation of a zygote and primary endosperm nucleus, and primary endosperm nucleus. Alright, so double fertilization leads to the formation of zygote and primary endosperm nucleus. This primary endosperm nucleus, this primary endosperm nucleus divides to form a large nutritive endosperm. This primary endosperm nucleus, which is made from the triple fusion, will undergo several mitotic divisions to form a large nutritive endosperm, which serves as, why this has been made, which serves as, so that it can serve nutrition, which can serve as the source of nutrition, for developing an embryo, all right. So to ensure that the developing embryo will have sufficient amount of nutrition, the zygote remains dormant in a fertilized ovule. So to ensure the nutrition for an embryo, the zygote remains dormant for some time, in a fertilized ovule. What happens actually when the fertilization takes place in flowers, in angiospermic plants, the double fertilization such as syngamy and triple fusion, which are occurring simultaneously inside the embryo sac of a flower. Alright, so what happens when syngamy occurs? It will lead to the formation of a zygote. Soon after becoming zygote, it will not undergo mitotic divisions to form an embryo. It will remain in that stage. The zygote will remain as a zygote only for some time, so that the primary endosperm nucleus, which is resulted from the fusion of three nucleuses such as triple fusion, can divide mitotically to form a large nutritive tissue, so that a developing embryo can have sufficient nutrition from that endosperm. That is why the zygote remains dormant, so that developing embryo can have sufficient amount of nutrition from endosperm. So this is the complete solution of question number 12. Now let's come towards question number 13, that is differentiate between. Basically in this question we have 4 parts, A, B, C and D. We have to differentiate between the given terms. So the first question or the first part in the question number 13 we have is to differentiate between hypocotyle and epicotyle. So what is the difference between hypocotyle and epicotyle? So it is the portion. If you are talking about hypocotyle, it is the portion present on the embryonic axis or embryonal axis just below the cotyledon in a dicot seed. It is the portion present on the embryonal axis just below the cotyledon in a dicot seed. And if we talk about the epicotyle, then epicotyle is also a portion present on the embryonal axis of dicotyledon seed, but it is present just above the cotyledons. Alright, so this is the basic difference between hypocotyle and epicotyle. Also, hypocotyle terminates into radicle and the epicotyle terminates into plumule. So if we write the difference between these two, in the left column we will be talking about hypocotyle, and in the right column we will be talking about epicotyle. The basic diagram of cotyledon and embryonal axis is just this kind of thing. These are the two cotyledons showing dicotyledon seeds. Now this is the embryonal axis. The portion below the cotyledons, in a dicotyledon seed on the embryonal axis represents hypocotyle. So this is the portion present on the embryonal axis just below the cotyledons, in a dicot seed is actually known as hypocotyle. And if we talk about the epicotyle, then this is the portion present on the embryonal axis lies just above the cotyledons. In a dicot seed. Also, the closing difference or the second difference between hypocotyle and epicotyle is that this hypocotyle region actually terminates into the region that is known as radicle, which will give rise to the root system of a plant. Alright, so this hypocotyle terminates into radicle. This terminates into radicle, which will give rise to root system of a plant, and this epicotyle terminates into the region known as plumule, and this plumule is going to give rise to shoot system of the plant. As you can see on the embryonal axis of a dicotyledon seed, you can see this is the region just above the cotyledon is known as epicotyle, whereas the region just below the cotyledon on the embryonal axis is known as hypocotyle. This hypocotyle is terminating into radicle, and this epicotyle is terminating into plumule, which will be giving rise to shoot system of a plant, and this radicle is giving rise to the root system of a plant. So this is the basic difference between hypocotyle and epicotyle. Now let's move on the second part of this question, that is differentiate between coleoptile and coleorhiza. So what is the difference between coleoptile and coleorhiza? This, if we talk about coleoptile, it is just the conical protective sheath. It is a conical protective sheath in case of monocot or monocotyledon seed, which is enclosing the plumule part. So this coleoptile, in the left column we are going to discuss about coleoptile, and in the right column we are going to discuss coleorhiza. This coleorhiza and coleoptile are the parts of monocot seeds, not dicot seeds. Alright, so what is the difference between coleoptile and coleorhiza? It is a conical protective sheath enclosing the plumule part of a monocot seed. Alright, and if we talk about the coleorhiza, it is also a protective layer enclosing the root cap and radicle in the monocotyledon seed. Alright, so this is the basic difference between coleoptile and coleorhiza, and these are the parts of monocot seeds only. So coleoptile is a conical protective sheath enclosing the plumule, of a monocot seed. Alright, coming on to the coleorhiza. What is coleorhiza? It is also a protective covering which is enclosing radicle as well as root cap in the monocot seed. So it is also a protective layer enclosing the radicle and root cap of a monocot seed. Alright, so it is also a protective layer which is covering or enclosing the radicle and root cap of a monocot seed. Why monocot seed? This is only present in monocot seed only. This is not present in dicot seed. Coleorhiza and coleoptile are the parts of monocot seed. They are not present as protective layers in dicot seed. So this is the basic difference between coleoptile and coleorhiza. Now moving on to the third part of question number 13, that is differentiate between integuments and testa. Now what is the difference between integuments? You must have heard about integuments, alright. What is integument? We know that we find integuments as the covering, the outermost covering of an ovule. What is ovule? The female megasporangium present inside the ovarian cavity or a locule. Alright. So this ovule is present inside the locule or an ovarian cavity. This ovule has hard coverings. This ovule has hard protective coverings, and these protective coverings of ovule is known as integuments. Alright. Now if we talk about the testa, testa is the outermost protective covering of a seed. Testa is just the outermost protective covering of a seed. Both are the coverings. Integuments is also a covering, testa is also a covering, but integument is a covering of ovule, that is female megasporangium, and testa is the covering of a seed after fertilization. Although this testa has been made from the integuments only. After fertilization, what happens in a flower or angiospermic plants? After fertilization, what happens to the ovule? That ovule along with its integuments convert into seed and its seed cover. The ovule, that is megasporangium of a female, converts into seeds. Ovule gets converted into seed after fertilization, and the ovary converts into fruit after fertilization. What happens with the integuments of an ovule? That integuments of an ovule converts into the protective layers, protective coverings of seed, that is testa. These integuments are going to convert into testa only. Alright. So the difference between testa and integument is, first we are going to talk about integuments. Now what are integuments? These are the outermost protective layers or coverings of a female megasporangium, that is ovule. That is ovule. We know that if we draw a diagram of an ovule, it looks like this. It looks like this. This having coverings. What are these coverings? What are these coverings? These are integuments only. These are integuments only. These integuments after fertilization along with the embryo sac, along with the ovule which is converting into seed, these integuments are converting into the seed coat which is known as testa. Now let's talk about the testa. What is testa? First of all, it is the seed coat. Seed coat or outermost covering of a seed. This is the outermost covering of a seed which is developed, from integuments only, from integuments only. After fertilization, the integuments convert into testa, which is the protective covering, hard covering seed coat. Alright, the covering of seed coat. So this is the basic difference between integuments and testa. Now moving on to the last part of this question, that is differentiate between perisperm and pericarp. Now we'll be talking about the perisperm and pericarp. First, we are going to discuss perisperm. What is perisperm? So, the persistent residual nucellus. Now, what is nucellus? We know that nucellus is the special kind of tissue which is present inside the ovule. Alright. So, what happens after fertilization? This nucellus tissue is actually digested by the developing embryo. In some seeds, it does not. It persists. It persists for time. Alright, so what is perisperm? The residual or the persistent residual nucellus in some seeds, that persistent nucellus is actually known as perisperm. It present in some seeds like beet or black pepper. Alright, so this persistent nucellus tissue in some seeds is known as perisperm, whereas if we talk about what is pericarp, it is the ripened wall of a fruit. Pericarp is just the ripened wall, outer covering of a fruit which is developed from the covering of ovary, alright, which is developed from the covering of ovary. Let's talk about perisperm. Perisperm here we are going to talk about. Then here in the column right we are going to talk about pericarp. Don't get confused with perisperm and pericarp, as both terms include peri. Alright, so this perisperm, if we talk about perisperm, then this is the persistent residual nucellus tissue. Alright, this is the definition of perisperm. What is perisperm? This is the persistent nucellus tissue in the seed which is known as perisperm. It is present in we are giving example of this. It is present in some seeds like beet and black pepper. Alright, black pepper. These are the examples inside which the seeds of black pepper and beet have residual nucellus, persistent nucellus in them. That is why we call them as perisperm, because after fertilization all things get changed, all things get converted into something. So similarly here, the nucellus tissue of an ovule is converting into perisperm, which is present in some of the seeds such as black seed and black pepper and beet. So this is perisperm. And if we talk about pericarp, this is the ripened, this is the ripened wall of a fruit. This is a ripened wall of a fruit. It is developed. It is developed from the wall of ovary. Because we know that after fertilization, ovary gets converted into fruit, ovule get converts into seed. As we know that the covering, the outermost covering of an ovule, which is known as integument, converts into testa, that is the covering of seed after fertilization. Similarly, in case of ovary, we know that ovary has a wall. After fertilization, when ovary converts into fruit, the wall of ovary also converted into the wall of fruit, that is known as pericarp. So this is all about the question number 13, and we have completed all the parts A, B, C and D, all the parts of question number 13. With this, we have completed the solution of question number 13. So now moving on to question number 14, and is asking why is apple called as a false fruit and which part of the flower forms the fruit? This is asking about apple only. First of all, to answer this question, we know that generally what happens after fertilization in angiospermic plants, what happens in after fertilization, the ovary converts into fruit. The ovary of a female converts into fruit and the ovule converts into seed. Alright, this is what happens generally. But in case of false fruit, or I may say that sometimes what happens, the fruit does not develop from ovary. The fruit does not get developed from ovary. These kind of fruits are known as false fruits. Apple is such an example of that kind of fruit. Apple is a false fruit. Why is it so? Because it is not developed from the ovary of a flower or a female organ. It is developed from thalamus. Which part of the flower which give rise or forms the apple fruit? Then the answer is thalamus. Thalamus is the part of a flower which give rise to the apple, or apple is developed from the thalamus. That is why, because apple is not developing from the ovary of a female organ, rather than it is developing from the thalamus of a flower. That is why the apple fruit is known as false fruit. Alright, so this is the answer, and if we are going to write, then it would be like this, that apple is a false fruit because it does not develop from the female ovary. Any fruit which is not getting developed from the ovary part, then the fruit is known as false fruit. Now moving on to the second part of this question, which part of the flower forms the fruit? Then the thalamus. The thalamus. Generally, what part of flower give rise to fruit? Generally it is ovary. Ovary gets converted into fruit generally after fertilization. But in case of false fruit in apple, the thalamus give rise to, the thalamus give rise to an apple. Alright. So this is the complete solution. This is the complete answer of question number 14. As we know that generally ovary gets converted into fruit, but in case of apple, thalamus is responsible for the formation of apple. Now let's talk about the question number 15. What is meant by emasculation. What do you mean by emasculation, and when and why does a plant breeder employ this technique? So first of all, this is emasculation. The emasculation is actually a technique employed by plant breeders during artificial hybridization. Alright, so this is a part of artificial hybridization. Now let's understand what is emasculation. As the name suggesting, emasculation means the removal, and masculation means the removal of a part of a male reproductive organ. When we remove a part of a female of a male reproductive organ, or we remove the male reproductive organ, that process is known as emasculation. So simply, the emasculation means the removal of a male anther or a male part from the flower is known as emasculation. When we remove anthers, this process of removing anthers from a flower is known as emasculation. Alright. Now, when and why does a plant breeder employ this technique? They do this in a bud stage. And why do they do so? Why these plant breeders emasculate the flower? They do so to prevent self-pollination. This is a very useful technique in case of artificial hybridization, when the pollen is required, when the desired pollen is required. Alright, that is why we do artificial hybridization, to prevent self-pollination and to obtain better quality seeds. Alright, so in the case of artificial hybridization, first we remove the anthers, that is known as emasculation, to prevent self-pollination. So let's just write the answer. Answer number 15. First of all, this emasculation is a technique useful in artificial hybridization. Alright, now what is emasculation? Emasculation, the process of removal of anthers of a bisexual flower, Now, plant breeders use this technique to prevent self-pollination and this is very useful in artificial hybridization, when the desired pollens are required to obtain the good quality of seeds. In case of artificial hybridization, this process is very useful to prevent pollination, to prevent self-pollination. Alright, so this is the complete solution of question number 15 here. Let's talk about the question number 16. If one can induce parthenocarpy through the application of growth substances, that means growth hormones of a plant, which is known as phytohormones. The hormones of a plant are known as phytohormones. Which fruit would you select to induce parthenocarpy and why? Alright, so to answer this, first of all, we have to know what is parthenocarpy. Parthenocarpy is the formation of fruit without fertilization. We know that in case of angiospermic plants or flowering plants, fruit forms after fertilization. After fertilization, the ovary gets converted into fruit, as well as simultaneously the ovule gets converted into seed. But in some cases, what happens, fertilization does not occur, but even then the fruit forms. So when fruit forms without fertilization, that process is known as parthenocarpy. And in case of parthenocarpy, that means when fruit is developing without fertilization, the fruit would be seedless. There is no formation of seed in case of parthenocarpy. And the fruits developed from this technique, parthenocarpy, are known as parthenocarpic fruits. Alright, now if we talk about the second part of this question, then what kind of substances or what kind of these things which can be induced into plants to obtain parthenocarpic fruits? So we know that first of all, parthenocarpy is a natural process. It can occur on its own in plants. Also, we can induce parthenocarpy with help of some phytohormones. The hormones of plants are known as phytohormones. So what is parthenocarpy? Parthenocarpy is the formation of fruit without fertilization. The seeds are known as parthenocarpic fruits, as well as they are seedless. They are not having seeds. These kind of fruits are known as parthenocarpic fruits. This parthenocarpy can occur on by own. That means it can be natural and it can be induced by some phytohormones. Alright, so it can be induced by some phytohormones. Next, if we talk about what fruits or what kind of things you can, which fruits would you choose to induce parthenocarpy? So, oranges, lemons, lychees, these are the fruits into which we can induce parthenocarpy. These lychees, oranges and lemons could be potential fruits to induce parthenocarpy, because these fruits, if we induce the parthenocarpy, there will be no seed in them and they will be more much appreciated if we induce parthenocarpy in oranges, lychees or lemons or any fruit with having seeds or so many seeds. If we induce parthenocarpy in these kind of fruits, then people will much appreciate it. People will much appreciate it, because after parthenocarpy, after inducing parthenocarpy into them, the fruit will be seedless and hence they are much appreciated by the consumers. This is why we induce the parthenocarpy thing into fruits. So let's write the answer of this. First of all, what is parthenocarpy? We will write parthenocarpy. So parthenocarpy is the formation of fruits without fertilization. Without fertilization. Alright, hence the fruits, because there is no fertilization, so the fruits will be seedless. The fruits will be seedless. There is no formation of seeds in them. Alright, it can be natural. Parthenocarpy can be natural and also can be induced in some fruits by some phytohormones, Alright, now what are the fruits in which we can induce parthenocarpy? That is oranges, lemons and lychees. Oranges, lemons, lychees could be potential fruits to induce parthenocarpy. Why is it so? Because these seedless varieties of fruits, the seedless varieties of these fruits would be much appreciated by consumers. Alright, so this is the reason why we induce parthenocarpy in some of the fruits, because they are much appreciated by consumers, and we can induce parthenocarpy artificially with the help of some phytohormones, or it can occur naturally in some of the fruits. I hope you get the answer of question number 16. This is the complete solution of it. Let's talk about question number 17, which is the second last question of this chapter. Explain the role of tapetum in the formation of pollen grain wall. So first of all, what is tapetum? Tapetum is actually the innermost, not outermost, it is innermost layer of the microsporangium. What is microsporangium? It is a sac-like structure present in the lobes of anther. Anther is a male reproductive part, alright. Stamen is the male reproductive part, which is composed of two things, that is anther and filament. Inside anther we find 4 microsporangias, and these microsporangias are lined by 4 type of layers. The innermost layer of the microsporangium is known as tapetum, which is responsible for providing nourishment to the developing pollen grain. As we know that pollen grains are developed inside the microsporangium. Alright. So tapetum is the innermost layer of the microsporangium, providing nourishment to the developing pollen grains. The cells of tapetum layer produces various enzymes, hormones, amino acids and other nutritive materials for the developing of pollen grains. Also, it produces the exine layer of pollen grain. The exine layer or the outer layer of pollen grain, which is known as exine, and is composed of sporopollenin substance. This sporopollenin substance is actually made or produced by the cells of tapetum layer. Alright, so this is the role of tapetum layer in the formation of pollen grain, or in the formation of the wall of pollen grain. This sporopollenin substance is actually highly resistant in nature to be ever known. Now, if you want to write the answer of question number 17, then it would be like this. First of all, we are going to write about tapetum. Tapetum is the innermost layer of microsporangium. Providing nourishment to the developing pollen grains. As we know that pollen grains are going to develop inside these microsporangium. So the tapetum is actually an innermost layer of the microsporangium, which provides nourishment to the developing pollen grain. The cells of tapetum produces various enzymes, hormones, amino acids and other nutritive material for developing or the development of pollen grain, for the development of pollen grain. All right, these cells also produces the exine layer of pollen grain, which is composed of sporopolanin, which is the highly resistant material in the nature ever known. Alright, so this is the tapetum, which is the innermost layer of microsporangium, which provides nourishment to the developing pollen grain. It produces various kind of enzymes, amino acids and hormones and all other nutrient material which can be useful for the development of pollen grains. Also it produces sporopolarin substance which is the highly resistant material in the nature, and the sporopolarin substance is actually making the exine layer or the outer layer of pollen grain to making it resistant in nature. So this is the complete solution of question number 17. Now let's come towards the last question of this chapter, question number 18, that is what is apomixis and what is its importance. So first of all we have to know that what is apomixis, then we are going to talk about the importance of apomixis. So first of all, apomixis is actually a mechanism of production of seed. Apomixis is a mechanism of production of seed without fertilization. When the seed produces without fertilization, that seed is known as apomixis. So that process is known as apomixis. This apomixis method is actually a type of asexual reproduction which mimics sexual reproduction. Alright, so this apomixis is a mechanism of formation of seeds without fertilization. This is a kind of asexual reproduction which mimics sexual reproduction, in which what happens, the female gametophyte, that is ovule, converts into an embryo. We know that ovule converted into seed. Here what happens, this ovule gets converted into an embryo, skipping meiotic divisions and syngamy as well. So apomixis is a method or a mechanism of production of seeds without fertilization. This is a type of asexual reproduction which mimics sexual reproduction, in which the female gametophyte or ovule gets converted or develops into an embryo, skipping meiotic divisions and syngamy as well. Thus, this apomixis produces the clonal progeny, clonal plants with the similar genotype of the mother plant. Alright, this is apomixis. Now, apomixis is of high importance as it produces seed. It produces the seed which are exactly similar to the mother plant. It produces actually the seed which are exactly have same genotype to the mother plant. Thus, it helps in the preservation of good characters over generation. It also helps in the formation of hybrid with a combination of desirable characters. It also helps in the formation of hybrids with the help of a combination of desirable characters. Thus, apomixis is helpful in preserving good characters. Also, it helps in the cost-effective and time-efficient production of seeds. So these are the importance of apomixis, and now we are going to write the answer of this. Question number 18th answer, apomixis. First of all, We are going to write apomixis is the mechanism of production of seed without fertilization. Apomixis is the mechanism of production of seed without fertilization. This is a type of asexual reproduction which mimics sexual reproduction, where the ovule, which is the female gametophyte, develops into an embryo, skipping meiosis and syngamy. Alright, so this is the definition of apomixis. Also it produces, because it is a type of asexual reproduction and there is no syngamy, there is no fusion of male and female gametes are taking place. So it produces the clonal progeny, with maternal genotype. Now come towards the importance of apomixis. So apomixis is of high importance. As it produces, so first is it produces the genetically similar seeds to the maternal plant or the mother plant. Thus apomixis is helpful in preservation of good characters over generation, Also it helps in preserving a specific character in a hybrid, as it is helpful in the production of seeds, in a production of hybrids, alright, with the help of a desirable character. So it is helpful in the preserving specific character in a hybrid. Last but not least, it is also helpful. It helps in cost effective and time efficient. Why time efficient? Because it is a process done in short time as compared to the sexual reproduction. So it is cost effective and time efficient of seed production. It is helpful and cost effective and time efficient in the production of seeds. So this is the complete solution of question number 18, and with this we have completed all the question in the textbook of NCERT, of your textbook of NCERT, and we have completed the chapter of sexual reproduction in flowering plants. So this is all about today. Thank you and have a nice day.